Hello, this is Eva Amson welcoming you to this Bite-Size Bio webinar, which today is sponsored by MilliporeSigma. MilliporeSigma is a leading supplier to the global life science industry with solutions and services for research, development and production of biotechnology and pharmaceutical drug therapies. Today's presentation is titled Sample Filtration in Pharmaceutical QC Testing, and it's being presented by Vivek Joshi from MilliporeSigma. Vivek is a senior R&D manager at MilliporeSigma, developing new products and applications for products that span analytical workflows, molecular and protein science workflows, and cell culture workflows. He has presented multiple posters and talks at conferences focused on filtration and membranes. Vivek is also a member of numerous expert panels within United States Pharmacopoeia, tasked with writing general chapters on filtration and various QC tests for pharmaceutical dosage forms. As always, we will have a question and answer session after the presentation. So please type any questions you have into the questions box, which appears on the top right panel of your screen, and I'll put them to Vivek at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email. So now over to you, Vivek, for the presentation.
Thank you for that introduction. So agenda for today's talk is that we are going to talk about importance of dissolution testing, followed by why is filtration critical in dissolution testing. We'll then talk about membrane characteristics and throughput in filtration. And finally, we'll wrap up the seminar with membrane properties that impact data. So let's focus on importance of dissolution testing within pharma QC. Dissolution is an important test that is carried out for all the oral drug formulations, and all samples are filtered prior to analysis. Anytime a drug manufacturer has to manufacture a formulation and release it to the market, dissolution testing is required as the QC test before the product can be released into the marketplace. Dissolution requires sample filtration prior to sample analysis, but there are a lot of other pharma QC tests like assay, content uniformity, stability, shelf life, particulate matter in injections, which also require filtration as the sample preparation step. When we think about dissolution testing and the drug development cycle, when we are looking at drug discovery phase, that's the time when we are looking at developing or discovering new compounds for a target molecule or a target analyte. At that point, there is no drug dissolution testing required. But as some of the hit molecules or the leads enter into the development phase, when we start working on formulation optimization or formulation changes, that's the point when dissolution test also starts getting developed. As these drugs move through the pipeline into the approval phase, when you are submitting your NDA, the local regulatory authorities require that dissolution testing is provided as the data package that is required for NDA submissions. Once the drug moves into the manufacturing phase, any manufacturing changes or process modifications. For example, if you decide to change the supplier for a particular excipient that you were using in your formulation, or for instance, you decided to make some manufacturing process changes, you have to show that the newly manufactured formulation still has very similar dissolution profile as the original formulation and the manufacturing process. Finally, as the novel chemical entities enter into the generic space, at that point, the goal of a dissolution test becomes providing bioequivalence to the innovator formulation that the company is working on. Pharmacopoeia are the governing bodies which regulate how the dissolution testing may be performed at this point. No matter what, whatever stage of a drug development cycle the dissolution test is performed, to generate useful information at each of these stages, dissolutions must be robust, easy to optimize and scale up, and produce consistent, accurate, and reproducible data. Now we'll go briefly over the dissolution testing workflow and some of the points to consider. As you are some of the experts in the dissolution working space or in the pharma QC space, I'm not going to go in detail about the dissolution testing workflow. but we'll focus on a couple areas where filtration can be critical. The first and foremost is the media preparation. Couple things to keep in mind when preparing your dissolution media is the water quality that is required for preparing media, as well as the amount of oxygen or air that is dissolved in the media. As you are all aware, the calibrator tablet that we typically use for calibrating dissolution baths, the prednisone tablet, its dissolution profile can be impacted by amount of dissolved oxygen that is present in the media. So it's critical to keep in mind that we deaerate or degas the media prior to using it for dissolution testing. And vacuum filtration using a membrane can be one of the simple methods for media deaeration. Once the media is prepared, depending on the type of formulation, whether it's an extended release formulation or immediate release formulation, you will carry out dissolution tests and collect samples at various time points. No matter what is the type of formulation that you are looking at, sample filtration has to be performed right after the sample is collected so that it can be analyzed by HPLC or LCMS. Most of the sample analysis in dissolution or any of the pharma QC tests is these days carried out using HPLC testing. For some of the older formulations, which only contain a single component, UV-Vis spectrophotometry might still be used. But majoritarily, HPLC is the means by which sample analysis is carried out. And here, the quality of samples and the mobile phase ingredients become very important. Now we talked about importance of dissolution test. Let's talk about why filtration is critical in dissolution testing. So you might ask, why do we need to filter a sample when we are doing your dissolution test or any of the pharma QC tests which require filtration? In dissolution test, the aim of the test is to find out how much of your API or the active substance has dissolved in the media at a certain time. In order to do that, we need to be able to separate the dissolved API or the active substance from the undissolved API as well as the excipients that may be present in the formulation. The simplest way to do that is by filtration. So essentially, filtration stops the dissolution process and provides us with a representative sample that is equivalent to the amount of API that is dissolved at that certain time point. There are two commonly used methods in the dissolution testing workflow for filtration of sample after the dissolution test is completed. The most common one is using syringe filters. About 70 to 80% of the customers use syringe filters. The Frit filters is another way of filtering sample prior to HPLC analysis post-dissolution test. In many times, these Frits may be present in your baths as part of your automated sample preparation techniques. Both the syringe filtration or the Frits can be used both in a manual format as well as in an automated sampling format. Let's compare the key differences or similarities between the syringe filters and Frit filters. The most important difference between the Frits and the syringe is their typical pore size. Normally, the syringe filters have much smaller pore size, anywhere from 0.1 micron to 1 micron. On the other hand, the Frits have much larger pore size, anywhere from 10 micron to 70 micron. So as you can imagine, if you are using Frits as your sample preparation or filtration technique post-dissolution test, the sample that you have filtered might still contain particles that are smaller than 10 micron in size. This might also include some of your undissolved API, if especially it is micronized. Or it might also include the excipients, which have much smaller size as well. Now you can imagine that these particles might clog up your HPLC column if the downstream analysis was done by HPLC testing. So even though the customers might be using Frit filters for their automated sample preparation technique, they might still have to filter samples through a 0.2 or a 0.45 micron syringe filter, depending on the downstream analysis. Other big difference between the Frit filters and syringe filters is their pore size rating. Syringe filters are typically rated by their largest pore, whereas the Frit filters have an average pore size rating. What that means is, in a syringe filter, when we say it is rated for 0.45 micron, the largest pore is typically 0.45 micron, and all the other pores will be smaller. On the other hand, when a Frit filter might have a pore size of 10 micron, it can have pores from anywhere from 5 micron to 15 or 20 micron, giving you an average pore size of 10 microns. So all in all, syringe filters definitely offer better removal of particles compared to the Frit filters. We know that filtration is critical for dissolution tests or many of the pharma QC tests. We also know that filtration stops your dissolution process. But when we ask a question to any of the pharma QC or the analytical development lab, the most common answer as to how they select their syringe filter is by what is available in the drawer or in the stockroom. Or sometimes they use the filter that worked last time in their application. Sometimes they talk about the filter being automation compatible. If you are a part of a QC lab, then you get a protocol that is created and validated by the methods development lab, and you really do not have a choice on selection of which filter do you use as part of your pharma QC test. But the key questions that we need to ask when selecting a filter is, what should be the pore size? For example, if you are doing HPLC as your downstream analysis, you should be using 0.45 micron syringe filters as your filtration choice. On the other hand, most of the UHPLC or UPLC vendors recommend that the sample be filtered through a 0.2 micron syringe filter before it gets injected. The second question that must be asked is the membrane material. The membrane has to be chemically compatible and also show low analyte binding as well as extractables. You should also think about the chemical compatibility of the membrane and the sample that you're trying to filter. And in some cases, where the samples have high level of particulates, it may have a pre-filter. So in the next few slides, we are going to talk about the membranes as well as their various characteristics that help you determine or select the right filtration product for your application. The first and foremost of the membrane characteristics that is very important when it comes to syringe filtration is the pore size. You have all seen syringe filters that have a pore size rating of 0.45 micron or 0.2 micron. And you might be wondering how this pore size is measured. These pores are so small that we cannot measure them by a ruler or micrometer screw gauge. The most common method for measuring pore size of a syringe filter or a membrane is by an indirect method known as bubble point. The way the bubble point test is carried out is using a membrane which is wetted with either water, if it's hydrophilic, or an organic solvent, if the membrane is hydrophobic. This wet membrane is held in place with one side exposed to the liquid or the wetting liquid, and on the other side, you have air which can be slowly pressurized. The air starts to get slowly pressurized, and there reaches a pressure at which the air is able to displace the liquid that is held in the membrane pores and comes from the other side in form of a stream of continuous bubbles. This is known as bubble point of a membrane. It is a pressure at which you start seeing a continuous stream of bubbles on one side of the membrane when the membrane is wetted. So typically, the bubble point is inversely proportional to pore size of a membrane. You can imagine that higher is the pore size, lower will be the pressure that will be required to allow for stream of bubbles to go from one side to other side. For example, a typical 0.45 micron membrane will have bubble point anywhere between 15 to 25 psi, depending on the membrane material and its manufacturing method. On the other hand, a typical 0.2 micron membrane will have bubble point in the range of 45 to 60 psi, again, depending on the membrane material and its manufacturing process. Some of the other methods for determining pore size is by bacterial challenge, specifically used when you are looking at sterilizing grade filters, or scanning electron microscopy or porosimetry are used when we are developing new membranes or membrane chemistries. When we think about membranes, we think that they may have very uniformly drilled holes, circular or cylindrical holes, drilled through the surface when we look at them. This can be completely different than what actually the membranes are, or how the porous structure in membranes is. For example, if we look at the PVDF or polyvinylidene fluoride membrane and look at the pore structure under a scanning electron micrograph, the pores are extremely random and they are interspersed with the polymeric material. Similar is the pore structure for a nylon as well as mixed cellulose ester membrane as well. All these membranes are manufactured by a technique known as solvent or air casting. And this technique creates pore structure, which is random in nature. If you look at the PTFE membrane, which is created using a heating and expansion process under pressure, you can see that the pores are expanded or stretched in one dimension because the membrane is formed using a PTFE film using heat and expansion technique. One of the very few membranes which have the uniform rounded or cylindrical pores is a polycarbonate membrane. The way this membrane is manufactured is by taking a polycarbonate film and irradiating it with electron beam. Following the irradiation, the membrane is then chemically etched, leaving behind cylindrical or circular pores where the membrane has been exposed to electron beam. We rarely use polycarbonate membrane in the pharma QC testing arena simply because they have very low lower number of pores, meaning they have a slow filtration rate. But polycarbonate membrane can be used when determining presence of particles in injectable formulations. So all in all, the manufacturing process of a membrane determines its pore structure and the morphology of membranes. Sometimes the membranes also may not have the same pore geometry across the thickness of a membrane. For example, as depicted in the cartoon on the top left, you can see that the Durapore or the PVDF membrane has a more symmetric pore structure, meaning that the pore structure is similar across the top and the bottom of the membrane. On the other hand, the polyether sulfone or a PES membrane has more asymmetric structure where the top surface has larger pores and the bottom surface has more or less smaller pores. The bottom area, which has the smaller pores, determines the particle retention behavior, whereas the top surface, where the pore size is larger, allows for faster filtration before the sample can clog the membrane. For instance, such type of a membrane or syringe filter might be useful when filtering samples which have high level of particulate impurities. One of the misconceptions that many customers have is all the membranes are created equal. For example, many times when we talk to customers, they say, oh, we have been using a PVDF or a PTFE membrane filter from one vendor, and we decided to switch to a PTFE or PVDF membrane with the same pore size from a different vendor. But we are now not getting the same performance as we saw earlier. one of the tests that we carried out recently with different PTFE membranes was retention of particles. In the left-hand bar chart, you can see that we tested the Millex hydrophilic PTFE membrane with 3 different PTFE-based syringe filters from different vendors. We filtered 0.5 micron particles through 0.45 micron syringe filters. As expected, you should see quantitative recovery or quantitative retention of these particles by 0.45 micron membrane. And the bar chart clearly shows that all the PTFE syringe filters that we tested, including the Millex, have the same high particle retention as expected. On the other hand, when we did the same testing using 4 different 0.2 micron syringe filters containing, again, the PTFE membrane, 3 of the 4 syringe filters showed the quantitative recovery as you expect for 0.24 micron particles. On the other hand, the PTFE syringe filter from one of the vendors only retained about 50% of the particles on the syringe filter. You might ask that, oh, these are all 0.2 micron syringe filters. Why is one particular membrane or why one particular syringe filter is not able to retain particles that are larger than the pore size? The reason might be the manufacturing process. The reason also might be how the membrane is rated or how the syringe filter is rated, how the pore size is determined. But don't simply assume that because you're keeping the membrane material same and the pore size same, the membranes will have the same behavior in different applications. The impact of this incomplete particle retention might be clearly seen in this example, where we actually ran a UPLC system with samples that were filtered through different syringe filters, as well as unfiltered sample and a sample filtered through a regenerated cellulose-based syringe filter. As you can see at the bottom, all the syringe filters that contain the PTFE membrane retained all the particles quantitatively. Thereby, there was no increase in back pressure, even up to 500 injections. The back pressure started off at about 4,000 PSI and ended up at about 4,000 PSI as well. On the other hand, as expected, when the sample was not filtered, it contained all the particulate impurity in it, and thereby the pressure kept increasing, starting from 4,000 PSI at the first injection to about 9,000 PSI at the 37th injection. We decided to stop this experiment at that point simply because we didn't want to break our UHPLC system. A regenerated membrane-based syringe filter performed slightly better than unfiltered sample, but still it allowed for significant amount of particles to go through into the filtered sample and showing an increase in back pressure as the number of injections went up. The regenerated cellulose filtered sample was able to run about 75 to 80 injections before the pressure reached the 8,000 PSI mark, at which point we stopped the experiment. So again, as I said in the previous slide, not all membranes are created equal, and there might be differences from vendor to vendor when selecting or switching from one vendor to another vendor. These days, many times our customers do not like to filter their mobile phase components. And I get it. Most of the time, we are purchasing our mobile phase solvents that are HPLC or UPLC certified. So many times, these solvents or water will be filtered through a 0.2 or a 0.1 micron filter. I understand that these solvents or the water will not have any particulate impurities when they are there in their original containers. But as we start making mixed mobile phases, or as we start mixing them together in labware or in the glass bottles that we have in the lab, you might start introducing particulate impurities. We did a similar experiment with the solvent filtration using different membranes from different vendors. For example, we filtered a 50-50 acetonitrile water mobile phase through hydrophilic PTFE membranes, as well as hydrophilic polypropylene membrane cut discs. We ran this mobile phase on a UPLC system overnight at 0.25 ml per minute. As you can see, when the solvents were filtered through hydrophilic PTFE membranes, the back pressure started at 3000 psi, and after 10 hours of run, the back pressure stayed about the same number, which is what is expected if the membrane is able to retain all the particles that are present in the mobile phase. On the other hand, when this mobile phase was filtered through polypropylene cut disc membranes, depending on the pore size of the membrane used, we saw that there was an increase in back pressure anywhere between 1500 to 2000 psi. Now imagine if you had samples that were filtered through a regenerated cellulose-based syringe filter that I showed in previous slide, and the mobile phase that was filtered through a polypropylene membrane, you can just get increased back pressure by incomplete retention of particles on both the membrane as well as on the syringe filters. Chemical compatibility of your syringe filter material is also equally important before selecting the type of membrane or solvent that you have. For example, most of the syringe filters are made out of either HDPE, that is polyethylene, or polypropylene. And across a wide range of chemicals, including organic solvents, aqueous basic solutions or acidic solutions, salt solutions, as well as surfactants, they have a very good chemical compatibility. When it comes to membrane materials, hydrophilic PTFE membrane has the broadest chemical compatibility across a wide range of chemicals. This includes organic solvents, aqueous solutions, salts, as well as surfactants. Followed by that, both PVDF and nylon membranes have similar chemical compatibility for a wider range of solvents. PVDF has better compatibility for aqueous solutions, whereas nylon tends to show slightly better chemical compatibility for organic solvents. The polyether sulfone and regenerated cellulose membranes generally have better chemical compatibility for aqueous solutions, but they may not work really well with organic solvents. So depending on the type of sample that you're filtering, you should choose the right membrane and the housing material for your syringe filters. Let's look at the physicochemical properties of membranes and its impact on the analyte binding characteristics. For example, in the top of this table, we have membranes like hydrophilic PTFE or hydrophilic PVDF or cellulose, which have very few functional groups, because of which they have very few interactions with your analyte of interest, thereby leading to low analyte binding. On the other hand, at the other end of the table, we have membranes like nylon or mixed cellulose ester, where you have large number of functional groups, which tend to show higher level of analyte binding interactions, thereby causing high level of analyte binding. For example, in case of nylon membrane, we have amino groups as well as carboxylic acid groups on either end of the membrane, followed by the various amide groups that are present as part of the backbone. All these functional groups can form electrostatic interactions as well as hydrogen bonding interactions. Think about it. If you have an analyte or API of interest, which is a basic molecule, under right circumstances, it will be charged depending on the pH of the solution. Similarly, under similar conditions, either the amino or the carboxylic acid group also might be charged, leading to electrostatic interactions as well as strong hydrogen bonding interactions with the amide bond. And that can lead to high level of analyte binding for nylon membrane. So generally, more functional groups mean more analyte binding. So when selecting a membrane, try and select a membrane which will tend to show low analyte binding to begin with. Moving on from the membrane characteristics, the next area to consider is throughput infiltration. Here, what is important is the device design or the filter design and its impact on how many samples can be filtered or how easy the sample filtration is. The first and foremost thing to keep in mind when selecting a filter is dependent on how much sample volume you're trying to filter. For example, if your sample volume was less than 1 ml, using a small sized filter is better, simply because it will have very low holdup volume, thereby you do not lose a lot of your sample. Also, because they have smaller surface area, they tend to show lower amount of analyte binding as well as low levels of extractables. On the other hand, if you are filtering anywhere between 10 to 100 ml of sample, a 25 or a 33 millimeter syringe filter might be better suited, because you might still lose about 100 to 200 microliter of your sample to the device holdup, but because of high filtration area, you will be able to filter the sample much faster without any discomfort. So the right size filter will improve throughput, reduce sample loss, but also minimize extractables and analyte binding. If you are filtering samples exclusively for HPLC, where the sample volume that you collect is only up to 2 ml, then a vacuum-based filtration system such as a Samplicity will allow you to filter up to 8 samples at the same time. With the Samplicity-based filtration system, you can use the same syringe filters, the 33 millimeter Mil-X filters that you have been using in your lab for manual filtration. But this takes away the syringe-based method for filtration and allows for filtration directly into HPLC vials. When it comes to difficult samples, one type of difficult samples or hard to filter samples are the samples that contain large level amount of particulate impurities. These are generally the samples which have or the formulations which contain high level of excipients which do not dissolve when the pharma QC test is carried out. What happens is the number of particles in these samples is so large that the particles clog the membrane that is used in the syringe filtration, thereby preventing you to get enough sample volume which can be collected or which is needed for downstream analysis. An easy solution for such type of a problem might be using a syringe filter which contains multiple layers of pre-filter followed by the final 0.45 micron or a 0.2 micron membrane. For instance, the HPF Mil-X syringe filter that we offer within our portfolio contains 2 layers of glass fiber pre-filters, the top one having a gradient in pore size starting from 10 micron to about 2 micron. Followed by that, there is another glass fiber pre-filter, which has a pore size of roughly 1 micron. Underneath this layer of pre-filters, you have your final 0.45 or a 0.2 micron membrane that is required for your final filtration of sample. What these glass fiber pre-filter layers do is they trap the particulates that are present in the sample, thereby preventing the membrane from getting clogged. And you can get higher amount of sample filtered with minimal effort. Here is an example using a protein solution where if you used just a membrane only filter, the filter clogs only after 1 to 1.5 ml of sample. On the other hand, if the same sample was filtered through an HPF Mil-X syringe filter, you can get up to 10 to 15 ml of sample before the membrane or the syringe filter clogs. So just by having a syringe filter, which contains an inbuilt pre-filter in it, you can improve your throughput significantly. The other type of hard to filter samples are samples which are viscous in nature. So they may not contain particulate impurities, but they generally have polymeric excipients, which when dissolved into your media, lead to an increase in viscosity. Now, Your HPF type of a syringe filter will not be able to remove these polymeric impurities from the sample simply because these polymers are dissolved in your media. In order to separate out these dissolved impurities from the dissolved low molecular weight API that you need for quantitation, what we recommend is using ultrafiltration membranes or ultrafiltration devices such as Amicon Ultra for separation of dissolved impurities. It contains a regenerated cellulose membrane, and they are rated not by their pore size, but by molecular weight cutoff. For example, anywhere from 5 kilodalton to 100 kilodalton for removal of broad range of polymers. These are typically used in protein concentration or DNA concentration, but they can be easily used for removal of polymeric excipients, thereby not only removing the particulate impurities, but also removing soluble impurities in form of polymeric excipients, which might also impact the downstream HPLC or UHPLC analysis. Finally, if you are doing automated filtration after dissolution test or after your pharma QC test, just make sure that the syringe filters that you are using are compatible with the automation equipment that you may have or the filter changing stations that you may have. Now let's talk about some of the key membrane properties that impact the data. Data accuracy and reproducibility is impacted by the chemical compatibility of the membrane with the solution that we need to filter. We talked about earlier that PTFE membrane has highest chemical compatibility, followed by polypropylene, PVDF, PES, and regenerated cellulose. The next two important characteristics of membranes filters or syringe filters is that filters exhibit variable drug binding. The drug binding is dependent on the physicochemical characteristics of the membrane, the analyte of interest, as well as concentration of analyte. Extractables are soluble materials that elute out of a device during filtration. These are generally low molecular weight impurities or low molecular weight polymeric impurities, and they can impact your downstream HPLC or UHPLC analysis. So let's talk about analyte binding. Here is an example of a dissolution test carried out using a 3-component migraine formulation. We did the dissolution test. Followed by that, we filtered the sample through 3 different Milex syringe filters, one containing PTFE membrane, one containing nylon membrane, and one containing PVDF membrane. We also collected different fractions through after filtration, going from 1st ml all the way to the 5th ml. As you can see, for the PTFE and the PVDF membrane-based syringe filters, there was no analyte binding observed even for the 1st ml of filtrate for acetaminophen. Whereas with nylon membrane or the nylon syringe filter, the 1st ml of filtrate only recovered about 58% of analyte of interest. As per USP, for an immediate release formulation, after dissolution test, the minimum concentration that the drug should be dissolved at is greater than or equal to 75%. By that requirement, this particular formulation would have failed for recovery of acetaminophen if we were using nylon syringe filter. Similar behavior was also observed for acetylsalicylic acid or aspirin, where with the nylon syringe filter, we only got about 30% of recovery for 1st ml of filtrate. On the other hand, with caffeine, which is a neutral molecule, even for the 1st ml of filtrate, we got quantitative recovery with nylon membrane. On the other hand, for PTFE and PVDF based syringe filters, for all the 3 components that were present in this formulation, we got quantitative recovery even for the 1st ml of filtrate. Here is an example where the analyte concentration has an impact on the drug recovery. We started off with about 250 ppm of naproxen and filtered the sample through 0.2 micron hydrophilic PTFE-based syringe filters. As you can see, for the first ml of filtrate, we get quantitative recovery for naproxen. But when the sample concentration was reduced to about 25 ppm or 2.5 ppm, the recovery dropped down by 3 to 4%. This might not be enough for it to fail the specification, but might be outside of the range based on the specification. As we go to the second or the 10th ml, quantitative recovery can be obtained for all the concentrations of API. So as you can imagine, when you have low concentration of analyte in your sample, that analyte might take more binding or may show more binding to the membrane before the membrane is fully saturated. In this example, we looked at the loratadine recovery after filtration, where we looked at 3 different hydrophilic PTFE membranes. I said earlier that generally hydrophilic PTFE or hydrophilic PVDF membranes tend to show low analyte binding overall. But in this case, what we observed was when we filtered the loratadine sample through 3 different PTFE syringe filters, the first ml of filtrate showed lower analyte recovery for all the 3 PTFE syringe filters tested. But by the second or by the 5th ml, quantitative recovery could be obtained for the Millipore hydrophilic PTFE syringe filter or a hydrophilic PTFE syringe filter from a different vendor. On the other hand, we observed that for one of the hydrophilic PTFE-based syringe filter, the filter did not saturate even after 5 ml of filtration. And we consistently saw only 80 to 85% recovery of this particular API or loratadine. This effect was even worse when the filtration was carried out using a 0.2 micron hydrophilic PTFE membrane. Again, the point to keep in mind here is that when we switch from one membrane to another or from one vendor to another, the behavior of different membranes might be different from different vendors. Here is an example of budesonide impurity profiling or an assay where we looked at filtration with PES, PTFE, and PVDF syringe filter. Again, I talked earlier that the PVDF and PTFE membranes generally tend to show low level of analyte binding across a wide range of chemical compounds. Here we could see that for the first ml of filtrate, both the PTFE or the PES membrane showed no binding and gave the expected recovery for budesonide from an assay standpoint. Whereas the PVDF membrane showed 30% analyte loss to membrane binding for the first ml of filtrate. But as we filter the sample further, from the second ml onward, quantitative recovery could be obtained using any of the 3 membranes. Here is an example of data that we obtained from one of our customers. For the formulation that they were testing, they were using a particular lot of PVDF syringe filters from us. And they were getting sample recovery of about 97%, which was slightly below their specification or toleration window, which meant that they had to carry out an out-of-spec investigation. We provided them with a different lot of a PVDF membrane, as well as we looked at a PTFE syringe filter. Both with the second lot of PVDF membrane and the PTFE membrane filters, we got quantitative recovery, which would have met their specification. When we started exploring this a little bit in detail, we realized that the customer was not filtering their standard using PTFE syringe filters. Even though this particular customer was discarding a fixed volume of sample before collecting it for analysis, the not filtration of standard was sufficient to cause a slight reduction in recovery when the sample was filtered through the PVDF-based syringe filter. As you can see, when the standard goes through the same process as the sample, the quantitative recovery could be obtained for all of the 3 syringe filtered lots that we tested. In your work, if you are looking or working in protein binding or protein analytes, then something to keep in mind is protein binding across various membranes is also quite different. Moving on from protein binding to extractables, here is an example of chromatograms from 3 different types of syringe filters from 3 different vendors. One containing hydrophilic PTFE membrane, two containing polypropylene syringe filters or polypropylene membranes, and the third one containing a nylon membrane. Mind you, all these syringe filters are certified for HPLC or UHPLC applications, indicating that they should have low levels of extractables. As you can see, the hydrophilic PTFE-based syringe filters do show very low level of extractables, as can be seen by really no peaks or very small peaks in the chromatogram. On the other hand, if you look at the chromatogram from a nylon syringe filter, where a sample of 70% acetonitrile and 30% water was filtered through the syringe filter, the filtrate was collected and analyzed using a reverse phase C18 column and 0 to 100% acetonitrile gradient. As you can see, the chromatogram shows a wide range of peaks in the time frame between 2 minutes all the way to up to 10 to 15 minutes. And also, this is the time in your chromatogram where your peak of interest or analyte peaks are expected to be eluting out. Similar peak profile was also observed, or extractable profile was also observed with the polypropylene-based syringe filters. The peaks that are observed here are not sharp as what we are seeing for the nylon-based syringe filter, but it's a broad peak indicating that there are some polymeric impurities that are eluting out in the sample and giving a broad peak in the chromatogram from an extractable standpoint. So all in all, keep in mind that all the HPLC certified syringe filters that you may have in your lab may not show exactly the same extractable profile when compared against one another. We talked about extractables being a problem. In order to get rid of extractables or to reduce the level of extractables in a syringe filter, it's very easy to just flush the syringe filter with your sample of choice or sample of interest. Herein shows a quick example where 0.01 normal HCl was filtered through nylon syringe filter, and the first and the second ml extracts were collected and analyzed by HPLC. As you can see, there are large number of peaks in the first ml of extract, but the level of peaks goes down significantly when we go to the second ml of extract. So you can imagine that if you discard, say, maybe 3 to 5 ml of the solvent or sample when filtered through a nylon syringe filter, you will get very low levels or almost negligible levels of extractables. Similar to what we see for HPLC chromatogram, here is an example of a polypropylene syringe filter using an LC-MS analysis. As you can see, there are a wide range of peaks that are showing up in the polypropylene extract that indicates that there are low molecular weight polymeric impurities that may be present in the polypropylene membrane that elute out when filtering the sample. Earlier, we talked about using ultrafiltration devices for filtration of highly viscous solutions. When these materials are used, or these types of filters are used for filtration of pharmaceutical samples, we also have to make sure that they do not introduce any extractable impurities. As you can see, the bottom, the pink plot shows that there are no or very low levels of extractable in a regenerated cellulose-based Amicon Ultra devices. On the other hand, the other type of ultrafiltration membrane, which is polyether sulfone, tends to show high level of extractables in the respective filtrates. Not only syringe filters, but even the syringes that we use can be cause of contamination when it comes to extractables. Here is again an example from a customer where the customer was trying to determine amount of zinc that is present in their formulation. They extracted the formulation with a concentrated mixture of sulfuric acid and nitric acid. When they filtered this sample through PVDF syringe filters, they saw that they were having high level of contamination of zinc. As the customer started evaluating where the contamination was coming from, they started with a plastic syringe and an air gap between the sample and the black piston seal at the bottom of the barrel. If there was no contact, the zinc contamination was fairly low, less than 10 ppb. On the other hand, when the sample started making contact with the black piston seal, the amount of zinc contamination went up from 96 ppb to 171 ppb. Where this zinc contamination might be coming from? These black piston seals are generally manufactured out of rubber, and many a times the rubber molders which make these parts might use zinc stearate as a mold release agent, and that may be the cause of zinc contamination. In this case, the customer even went with a glass syringe and a metal lure fitting and a PTFE piston to get rid of the black piston seal. And not only they saw a higher level of zinc contamination, almost 470 ppb, but they also saw contaminations coming from nickel, chromium, manganese, which are part of the stainless steel lure fitting that the customer had used. So all in all, any surface that comes in contact with the sample has potential to introduce extractables as well as cause analyte binding. So in summary, filtration is very simple sample preparation technique, but it's very little understood. The three key points from this seminar would be hydrophilic PTFE membranes tend to have the broadest chemical compatibility and very low levels of extractables as well as analyte binding. The second membrane to keep in mind when thinking about filtration is hydrophilic PVDF membrane, which is generally compatible with a broad range of aqueous solutions. It also has low extractables and drug binding and is recommended for protein analytes. Finally, selecting filtration format based on the throughput needs will help improve your filtration overall. With that, I would like to wrap up the seminar, and if there are any questions, I will be more than happy to take them. Thank you.
Thank you, Vivek. That was an excellent presentation. We've got a few questions from the audience already, but if anyone else has a question, please post this in the questions box that appears on the top right of your screen. So the first question that we've got for you here is, when can we use centrifuge?
So centrifugation is not considered as a preferred method of sample prep following any of the pharmaceutical QC testing, simply because even though the centrifuge or the centrifugation will settle the particles at the bottom, it does not actually remove the particles. So when you are pulling out your sample for analysis, you might disturb the bottom particle layer and may pull those particles into your sample. The only time I would recommend using centrifugation is if you are concerned that there is some kind of an analyte binding to your filter and you want to prove whether it is coming from the filtration or the syringe filter device. You might use centrifugation as kind of a control to show that there was no analyte binding happening. But that's the only scenario where I would recommend centrifugation as means of separating particles.
Thanks. That makes sense. And what filters are recommended for viscous products?
So for viscous products, what we recommend is filters that contain ultrafiltration membrane, like the Amicon Ultra that I showed in the presentation. When you have a viscous product, what it means is you have some kind of contaminants or impurities in them which are dissolved. Generally, these are the polymeric impurities or polymeric excipients, which when dissolved in the media, lead to increase in viscosity of solution. So you may or may not have high level of particles, but you have a difficult sample to filter. In this case, using an ultrafiltration membrane, you are able to remove these dissolved or solubilized polymeric impurities based on their molecular weight. So for example, if you have a polymeric impurity that has a molecular weight of about, say, 20 or 30 kilodalton, then you can use a 10 kilodalton cutoff ultrafiltration membrane or ultrafiltration membrane-based device to remove those polymeric impurities, and at the same time also get rid of the particles that may be present.
Thanks. Those are all great suggestions. This was another question that came in asking you for advice. Because most of the dissolution mediums contain aqueous solution, we use a glass filter. Can you advise?
Sure. So typical glass fiber membranes or glass filtration devices, they have a pore size of 1 micron. If you are doing HPLC or UHPLC downstream, the normal recommendation for filtration is either through a 0.45 micron membrane or a 0.2 micron membrane. So if you are using a glass fiber as your filtration membrane, you may not be able to remove all the particles that may be present in your sample, which can cause clogging of your HPLC or UHPLC system. Another disadvantage with glass fiber membrane is that they tend to have higher analyte binding as well as higher levels of extractables. So you just have to keep that in mind if you are using glass fiber as your membrane.
Thanks. And a question that just came in is, which filters do you recommend for buffer solutions used as mobile phase in HPLC?
For buffer solutions, you can either select a hydrophilic PTFE membrane or a hydrophilic PVDF membrane for your filtration application. Both of these membranes will have generally low analyte binding, low extractable impurities, as well as they are compatible with your aqueous buffer solutions.
Thanks. I've just got a few questions left, unless we get any more questions in. How can you reduce the level of extractables in a syringe filter?
In order to reduce the level of extractables in a syringe filter, the easiest way is, one, you start with a membrane or a syringe filter that inherently has low levels of extractables, like the hydrophilic PTFE or PVDF membrane. But if you end up with a membrane that tends to have higher level of extractables, what you can do is by flushing the membrane with your sample. So what that does is, as you flush your syringe filter or membrane with your sample, the extractables that are low molecular weight soluble impurities will get washed off the membrane, thereby reducing the level of extractables. If I recall, I showed it in one of my slides where the level of extractables in nylon membrane can just be reduced by instead of collecting the first ml fraction, if you collected the second ml fraction or the third or the fifth ml, you can reduce the level of extractables significantly.
Thanks. And that just leaves me with one final question. Is sample filtration the only type of filtration needed to remove particles before HPLC?
Yes, sample filtration is the only type of filtration that is needed to remove particles. Whether you use an ultrafiltration membrane or a microporous like a 0.2 or a 0.45 micron membrane, those membranes are the ones that are going to remove particulate impurities from your sample. so that you can do downstream HPLC or UHPLC analysis.
Great. Thank you so much. That brings us to the end of our webinar today. So thank you again, Vivek, for a very illuminating presentation and a great discussion. And
thank you, Eva.
Yeah, thanks, of course, to our sponsor, Merck KGaA, Darmstadt, Germany. And finally, thanks to the audience for taking the time to attend and listen in. So until next time, good luck in your research and goodbye from all of us at Merck and Bite Size Bio.